So we're continuing in Chapter 8, and here we're going to focus on transcription and translation, or RNA and protein synthesis. So RNA is ribonucleic acid, so it's very similar to DNA, just that slight difference of the oxygen. And other differences that you find with RNA is that it occurs as single-stranded rather than double-stranded like DNA. It is the ribose sugar, but again, DNA is deoxyribose. This is just ribose. And then in the sequence of RNA, you have U's instead of T's. So RNA sequences are like A, U, G, C, C, A, U, U. There's no T's. And there's three different types of RNA. There's ribosomal RNA or rRNA, which makes up ribosomes, transfer RNA or tRNA, which is involved in transporting amino acids to the ribosome during protein synthesis, and messenger RNA or mRNA, which is the one that is actually translated into protein. But before we get to translation, to talk about transcription. So transcription is the process by which the DNA sequence is converted into an RNA sequence, and or the process by which RNA is made, right? So it's not just mRNA, but tRNA and rRNA are also made in the processes of transcription, but we do focus on mRNA because it is the one that will get translated into a protein. So transcription begins on the DNA at an area called the promoter. That's where it kind of marks that that's where the gene starts. So it is important to distinguish, unlike DNA replication, where the entire chromosome is getting replicated, here it's not the entire chromosome, it's just the gene segment that gets transcribed. So that's where the promoter marks the start of the gene, the terminator marks the end of the gene. Transcription is done by the enzyme RNA polymerase, and just like DNA polymerase, it can only synthesize in the 5' to 3' direction. So we can see that here. So here's in pink the promoter sequence where the gene begins. This is RNA polymerase here, this light blue circle. So RNA polymerase binds at the promoter. The DNA gets opened up a little bit right there to allow for the enzymes to come in. The RNA polymerase moves along the DNA, reading the DNA sequence and synthesizing this RNA transcript, which is shown here in green. So it's not creating a double-stranded molecule. It's just creating a single strand of RNA that is based on one of the DNA strand sequences. That DNA strand sequence is called the template strand. It's the bottom strand here. And you can see that it's just based on complementary base pairing. In the DNA, whenever there's an A, remember there's no T's in RNA, so there's a U across from it. But wherever there's a T in the DNA, then the RNA has an A. G's and C's are still the same. So the only difference is anywhere where you would want to write a T across from that A, instead of a T, it's a U. So then transcription proceeds until the terminator sequence is reached, and then RNA polymerase removes from the DNA, and the RNA transcript gets released. And if that is mRNA, then it's going to go on to get translated. If it's tRNA or rRNA, they're going to travel over to ribosomes to do their jobs of translation.
Cells use DNA to store information, while proteins are used to perform the activities necessary to keep the cell alive. In a process called transcription, the information stored in a DNA molecule is copied into RNA molecules, which can be used to synthesize specific proteins. Cells use three types of RNA: ribosomal RNA molecules, which are used to form ribosomes; transfer RNA molecules, which deliver amino acids to ribosomes; and messenger RNA molecules, which carry the information for making specific proteins from DNA to ribosomes. There are three stages in the process of transcription, initiation, elongation, and termination. Let's see how each stage works. During initiation, RNA polymerase binds to the DNA and recognizes a site called a promoter at the 3' end of the template strand of the target gene. When RNA polymerase binds a promoter, it breaks the hydrogen bonds holding the DNA strands together at the site of the promoter, and transcription begins. RNA polymerase does not bind to all promoters with equal affinity. This difference in promoter strength is one way that cells can control gene expression. The more strongly RNA polymerase binds to a particular promoter, the more likely that gene is to be transcribed. In general, higher levels of transcription lead to higher levels of translation, which leads to higher concentrations of that polypeptide. One strand of the DNA serves as a template strand. The RNA transcript is copied from this strand of the gene, and therefore has an RNA sequence complementary to this strand. During elongation, RNA polymerase moves along the DNA, opening up a bubble in the DNA. As it moves, RNA polymerase adds bases to the 3' end of the growing RNA transcript. Elongation continues until RNA polymerase reaches a site on the DNA called a terminator. At this site, RNA polymerase and the newly synthesized RNA transcript are released from the DNA. Termination can occur through two mechanisms: self-termination and enzyme-dependent termination. In self-termination, the RNA sequence transcribed at the terminator causes the RNA to hydrogen bond with itself, forming a stem loop structure which essentially pulls the RNA polymerase off the DNA. In enzyme-dependent termination, a termination protein binds to the terminator and pushes RNA polymerase off the DNA.
Okay, so that was transcription. Moving on to translation. So translation is when the mRNA is now turned into the language of proteins. You might have heard in the animation he said polypeptide. So you can kind of, for us anyhow, you can kind of use peptide, polypeptide, and protein interchangeably. I mean, there are some differences. Peptide is just a sequence of amino acids, and that's what initially translation makes. The mRNA is turned into or translated into a sequence of amino acids, a peptide. Poly just means many. So polypeptide is more chains or longer chains sometimes of amino acids, of peptides, because peptide usually refers to kind of a small chain. And then a protein is where those polypeptides have come together, sometimes multiple of them bound together and folded up into a three-dimensional form to actually create a functional protein. But again, for us, we're not really going to distinguish peptide, polypeptide, proteins. So any of those terms, just know that that is what is made in translation. And of course, proteins are what then carry out the functions and give the unique characteristics of organisms. And that's all encoded in that organism's DNA. And it's expressed, the protein is created through the processes of transcription and translation. So how does mRNA get turned into sequences of amino acids, into a protein? Well, it's through codons. Codons are groups of 3 nucleotides in the RNA that code for a particular amino acid. And so there's a genetic code, and it's universal. Bacteria have and use the same genetic code that we do, that plants do, that mosquitoes do. All living organisms use the same genetic code. And this genetic code, the way it works is there's 61 what we call sense codons, meaning that they code for an amino acid. They code for 20 amino acids. So there's a lot of duplication, or what we call redundancy, where there's more codons than there is amino acids. So multiple codons will code for the same amino acid. And we'll see that in a minute. And then there's degeneracy, which kind of is the same thing, I guess. Redundancy, degeneracy, however you want to call it. Again, just meaning that each amino acid is coded by several codons. But this degeneracy or this redundancy is good because we're going to see when we talk about mutations that it does help to prevent that all mutations will be harmful. And we'll see that in a little bit. But so translation always begins at the start codon. The start codon is AUG. Translation ends at one of three nonsense or stop codons, either UAA, UAG, or UGA. The codons in the mRNA are read sequentially, just like the DNA was read to make RNA, and the RNA is now going to be read to make the protein. So this is the genetic code table. So it might look a little intimidating at first, but it's really quite easy and straightforward, and you do not have to memorize it, okay? You will be expected to be able to use it, and it will be provided on the exam for you, okay? So here is the, if we just start in this upper left-hand corner, here is codons that start with U. UUU, UUC, UUA, UUG. You notice that these two code for this amino acid, which is phenylalanine. I would not expect that you know that. Just using PHE would be fine. These two codons, as well as these four below, they all code for the amino acid leucine, LEU. So you can see there's 6 codons for leucine. Here there's 3 for isoleucine, ILE. Here's that start codon. It's only 1 codon for methionine, which is always the first amino acid. And so on. Going through the genetic code, you can see that degeneracy or that redundancy, that there is multiple codons for the same amino acid. What you may also notice is that it's the third base that often is the only difference, right? CCU, CCC, CCA, CCG. We call that wobble, meaning that this third base can wobble. It could switch between any of the other nucleotides, and it doesn't matter. If a mutation happened that affects that third base, it doesn't matter. It's still going to indicate proline, and it's not going to have a negative impact on that organism that has experienced that mutation. Okay, so translate the following sequence into amino acids. So what you have to do here is first realize that this is a DNA sequence. How do we know it's a DNA sequence? It has T's, right? RNA does not have T's, RNA has U's. So what's the first thing you have to do if you want to translate? Well, we first have to transcribe. So first thing you want to do is take this DNA sequence and turn it into RNA. Okay, and it's just based on base pair ruling. So I'll get you started and then you'll answer the rest in the questions. So what would go across from a G? C. C, G. A. Do we put a T? No, because we're translating. So, well, we're transcribing first and then translating, right? But we're making RNA. So does RNA have T's? No. So what's going to go there? A U. So U. So it's C, G, U, U, G, C, A, right? So across from a T is still an A. The difference is that RNA strand that you're going to synthesize here, it just won't have any T's. So anytime across from an A, you have a U instead. But across from a T, it's still going to be an A there, okay? So we'll see that in the question. So that's the RNA. I'll probably have to break the question into 2 parts. So you'll have one where you make the RNA, and then you'll have one where you actually translate into amino acids. That's a little trickier. So let's also just do part of this. Okay, so we saw that here the first codon, the first triplet codon, right? If you divide this into 3s, would be C, G, U. right? CGU. So what amino acid is that? Well, that's where you have to use the genetic code table. So first position C, second position G. So we know it's going to be in this box here. And there it is, CGU, arginine. Okay, so you do that for each triplet. ACG is going to be UGC. So again, go back here, first position U, second position G, and then just look for the UGC, cis or cysteine. Okay, so again, this will be kind of a two-part question that you will answer. So in the process of translation, the tRNA molecules carry in the amino acids over to the ribosome because translation occurs on a ribosome. And how does the tRNA know what amino acid to bring in? Well, the tRNAs have what's called an anticodon that is complementary to the codon on the mRNA. So let's say, if you're actually starting with AUG, as it should, right? So the start codon on this protein that's being made, on the mRNA, it will be AUG. The amino acid is methionine. So there's a tRNA that has an anticodon to this codon, and that's how it knows to carry methionine. So what would be that anticodon on that tRNA? Well, it would be complementary to this codon. So again, it's just based on, in this case, it's RNA, so A's and U's and C's and G's. So A's and U's, so it would be U, A, C, right? That would be the anticodon that the tRNA has that is carrying methionine. So it knows to carry methionine. What about in our example here? So our codon was CGU. So let's go back and look at CGU was over here, right? It was arginine. So the tRNA that is carrying the arginine is going to have the anticodon to that. So what is the tRNA going to have? It's going to have GCA. Right? GCA. So again, it's just based on that complementary base pairing. So here's a diagram showing the process of translation. So translation occurs on ribosomes. Ribosomes are made of two subunits, the large subunit and the small subunit. You might remember that for prokaryotic or bacterial ribosomes, they are 70S. They have a 50S and a 30S subunit. So that's what we're looking at here, the 50 and the 30. Here's your start codon, AUG, on the mRNA, right? And here's your tRNA with the appropriate anticodon carrying the appropriate amino acid, methionine, okay? Then the ribosome, so it assembles at the start codon. The tRNA brings over the amino acid, again, based on that anticodon sequence, that complementary base pairing. And then the ribosome moves along the mRNA. So the next codon it comes across is UUA. And so the tRNA with that anticodon would be AAU. And again, if we went over to the genetic code table, you would see that UUA is leucine. And it continues. The ribosome moves along the mRNA, reading each codon, and there's a tRNA with the corresponding anticodon that's going to then bring in the corresponding amino acid. Don't worry about the A site, the E site, the P site. We really don't need to get into the details of that. But basically, it's just different locations on the ribosome where, different things are happening here with the tRNAs and the amino acids. But basically, big picture is these amino acids get joined together with peptide bonds, thus forming a peptide, a chain of amino acids that eventually forms a protein. Right, so the process just continues, getting this growing polypeptide chain. And the process ends when a stop codon is reached, also called a nonsense codon, because it doesn't code for anything. There's no tRNA with the corresponding anticodon because there's no amino acid for the stop codons. So the ribosome knows when a stop codon is reached and there's no tRNA to bring in anything, it knows the process is done. The ribosome will remove itself from the mRNA. The polypeptide will get released, as you see here. And then once it folds up into its proper configuration, that's when it's actually called a protein. So what's interesting is that in bacteria, you can actually have simultaneous transcription and translation happening at the same time. And this was captured on an electron micrograph. You can see here. This very faint yellow string here is the DNA. And then you can't see the string of the RNA, but you see the red. The red is ribosomes that are bound all over the RNA, and they're making a protein, which you also cannot see in the picture. So what they've done is they've shown it, drawn it here. So the purple is the DNA. This kind of teal is the RNA that's being transcribed off of the DNA. And even before transcription is done, ribosomes are jumping onto the start codon and making peptides. They're doing translation at the same time. So this explains why bacteria can replicate so quickly. They can do simultaneous transcription, translation. They can make proteins very quickly to carry out all their cellular processes. What about us? Why can't we do simultaneous transcription and translation for any other eukaryotic cells? Well, it has to do with the cells and the nucleus, right? So in bacteria, in prokaryotes, there's no nucleus. The DNA is just right there in the cytoplasm, and so are all these enzymes and ribosomes and everything. It's all in one location, so it can happen. But in eukaryotic cells that have their DNA in a nucleus, transcription occurs in the nucleus. But where are ribosomes? Are there ribosomes in the nucleus? No, they're in the cytoplasm. So what has to happen is the mRNA gets made in the nucleus. It gets sent out of the nucleus, and then transcription. So transcription occurs only in the nucleus, and then translation occurs in the cytoplasm. So you can never get simultaneous transcription translation in eukaryotes because of the nucleus. It divides up where those things happen. But prokaryotes, because they don't have a nucleus, you don't have that problem, and you can get this simultaneous transcription and translation. Another quick thing to point out, I don't really want to get into the details of it, but transcription in eukaryotes is more complicated because the transcript has to be processed. It has to have non-coding DNA that gets cut out, which are called introns, and then the exons are spliced together. You may have learned about this before. I'm not worried about it because guess what? Bacteria are simpler. That's why I like them. That's why I like microbiology. We don't have to talk about exons and introns because bacteria don't have them.
Cells use DNA to store information, while proteins are used to perform the activities necessary to keep the cell alive. In a process called transcription, the information stored in a DNA molecule is copied into RNA molecules, which can be used to synthesize specific proteins. Cells use three types of RNA: ribosomal RNA molecules, which are used to form ribosomes; transfer RNA molecules, which deliver amino acids to ribosomes; and messenger RNA molecules, which carry the information for making specific proteins from DNA to ribosomes. The 4 nucleotides of messenger RNA are arranged in 64 three-letter words called codons. The genetic code is a language consisting of these 64 codons. 61 of the 64 codons code for a specific amino acid, while the remaining 3 instruct the ribosome to stop the translation process. Let's see how this table is used to determine the amino acid of a given codon, CAG. First, on the left side of the table, find the first letter of the codon, C. Then on the top of the table, find the second letter of the codon, A. Finally, locate the codon that has a G as its last letter, and you will see that the amino acid coded for by CAG is glutamine. Can you determine which amino acid is coded for by UCA? Serine is correct. Now, can you determine the effect of UAA? The codon UAA signals termination of the elongation of the polypeptide. It's a stop codon. There are 3 stages in the process of transcription: initiation, elongation, and termination. Let's see how each stage works. During initiation, RNA polymerase binds to the DNA and recognizes a site called a promoter at the 3' end of the template strand of the target gene.
Hold on. Okay. This was a mistake. This was supposed to be translation, and the wrong animation is in here, and we already watched this one, so it's fine. We really don't need all the details of that animation for translation anyhow. If you want more, go to the animation and videos that I have in Canvas. I think I have a good Amoeba Sisters one on translation that you would enjoy. So I would look at that instead, because I know the one that I should have here in the PowerPoint is way more detailed. It goes into the A site, the E site, the P site, which you're not going to be responsible for. All right, so almost another 30 minutes has went by in this part 2 recording, and so I'm going to stop the recording here, and part 3 will pick up with mutation, and hopefully we'll be able to finish out this chapter in part 3.